Oops. All right, so let's start with part 2 here in chapter 7. So again, glycolysis basically means you're breaking down, splitting glucose, so sugar splitting. It's going to break down glucose into 2 molecules of pyruvate. You're going to get 2 water molecules as a byproduct. And this occurs in the cytoplasm, and you have 2 major phases. You have the energy investment phase and the energy, your book calls it the energy payoff phase. It's also called the energy harvest phase. Okay. So the net yield of energy here is going to be 2 ATP molecules plus 2 NADHs per glucose. Now, what we mean by net energy is you really make 4 ATP at the end of this, but in the energy investment phase, we got to put 2 energy in to start the process. Okay, so that's what that's talking about. So you need to know glycolysis has 2 major phases: energy investment, energy payoff, and the net yield, 2 ATP, 2 NADH. All right. Now, glycolysis is going to occur whether or not oxygen is present. If oxygen is present, the energy that is stored in the pyruvate and the NADH will then go on through pyruvate oxidation to the citric acid cycle and oxidative phosphorylation. So make sure you watch that glucose or glycolysis in the study area. If we don't have oxygen present, you just undergo glycolysis. So this is why having oxygen is much more efficient. If our bodies had to just undergo glycolysis, we'd only be making 2 ATP for every glucose, whereas with oxidative phosphorylation, citric acid cycle, and glycolysis, we could be making 36. So 36 is a heck of a lot more energy than 2. All right. So again, glycolysis, we're going to get some ATP. And again, this occurs in the cytoplasm or the cytosol. We don't need oxygen. All right. So this is a whole series of steps. I don't want you to know every step. All right. Let me preface that. You do not need to know every step. This is part 1, the energy investment phase. Okay, what I want you to know is you start with 1 glucose molecule. It's going to go through a series of reactions. In order to get those reactions to occur, we have to use ATP twice. We have to add 2 energy to get this to occur. Okay? And at the end of the energy investment phase, remember, glucose has 6 carbons. It's C6H12O6. We're going to have a whole bunch of enzymes. We're going to add 2 ATP to give energy to start this process. And at the end of the energy investment phase, you're going to have 2 G3Ps, glyceraldehyde 3-phosphates, and you're going to have some of this DHAP. So they can float back and forth between the 2 of them, but it never really equals equilibrium, because as soon as G3P is formed, it goes to the next phase. So, takeaway points. Glycolysis, energy investment phase. 1 glucose goes through a series of 4 or 5 reactions. We add 2 ATP to start those reactions. At the end of the energy investment phase, you have 2 glyceraldehydes. Notice that the glyceraldehyde, the G3P, and it's glyceraldehyde because it has the aldehyde functional group, and 3-phosphate because the phosphate is on the 3rd carbon. Anyway, 1 glucose, you had 2 ATP to start it, a series of reactions. We end up with 2 3-carbon molecules called G3P. That's what I want you to know from the end of this, okay? All right, now here's the energy payoff, or what we call the energy harvest phase. You have the 3 G3Ps, okay? And again, notice that there are 5 or 6 reactions here. You don't need to know all of them. All I want you to know is that G3P is going to get converted through a series of reactions, and as that occurs, you're going to reduce NAD plus to get 2 NADHs, because you're going to take a hydrogen off one of each G3P, so that's why you got 2 of them, because you got 2 G3Ps. And as it goes through a series of reactions, you're going to make 4 ATP. You give off some water here, and at the end of this process, you have pyruvate. So at the end of the energy payoff phase, 2 G3Ps get converted through a series of reactions to 2 pyruvates, and in the process, you're going to make 2 NADHs, 4 ATP, and you're going to give off 2 water molecules. That's what you need to know. Okay? So, here, I put a little yellow box here. You need to be able to tell me this summary on a test. So again, notice we made 4 ATP, but it's like buying a lottery ticket. If you spent $2 on a lottery ticket in the energy investment phase on a scratch-off, and you won $4, you're only ahead $2, okay? So, we're only making overall 2 ATP because we had to put 2 in. We used 2 up to begin with, so we were in the hole, and we made 4, okay? So, energy investment phase, glucose, you got the 2 ATP is used to make this 2 ADP and the 2 phosphates. You got the G3P. The energy payoff phase, we had 4 ATPs formed. We make 2 NADHs, 2 pyruvates, and 2 water, and here's the net equation. So make sure you can tell me this. If you can tell me this, you'll be good as gold. You do not need to know every single specific step. All right. After you make the pyruvate, if we have oxygen, only if we have oxygen, it'll go to the mitochondria to complete its oxidation. And when it's in the mitochondria, here's where we're going to get some of the carbon dioxide that's released. So carbon dioxide is going to be released. The pyruvate is converted to acetyl coenzyme A, which we call acetyl-CoA. And then that acetyl-CoA is going to go into the citric acid cycle. Okay? So, by breaking down pyruvate, you get 1 NADH per pyruvate. So you get 2 NADHs per glucose molecule. Because remember, glucose has 6 carbons. It's split into 2 3-carbon molecules called G3P. Those 2 G3P are converted to 2 3-carbon molecules called pyruvate. So that's why we're getting 2 of these things. All right. Citric acid cycle is also called the Krebs cycle, depending on your book. That's going to complete the breakdown of the pyruvate to carbon dioxide. So this is where all of our carbon dioxide's coming off, or most of our carbon dioxide. So each turn of the Krebs cycle oxidizes 1 pyruvate. So it has to go 2 times. Okay? So when you see a diagram, if it only says 1 ATP, 3 and 8, you have to double it. So it takes 1 complete circle of the Krebs cycle to oxidize 1 pyruvate. Krebs cycle happens twice to completely oxidize the pyruvate. At the end of the Krebs cycle, or the citric acid cycle, I'm old school here, you're going to get 2 ATP, 6 NADHs, and 2 FADHs, FADH2, that should say FADH2 per glucose molecule. Okay, make sure you watch that animation, the citric acid cycle. All right, so glycolysis has occurred. We've made our 2 ATP, okay, but you're running a marathon. You have a lot of oxygen in your muscle and your blood, so the pyruvate's going to undergo oxidation into the citric acid cycle. We're going to make a couple more ATP. That's what we're getting ready to talk about. All right. Again, make sure you can summarize this for me on a test. All I want you to be able to tell me is pyruvate that comes from glycolysis. Remember, you have two of them. Is going to go through a series of oxidations where you give off carbon dioxide, you make an NADH, and you make an acetyl-CoA. Okay? So notice that now the acetyl-CoA only has 2 carbons because you lost 1 carbon in a carbon dioxide. Okay? Now, for the citric acid cycle, you do not need to tell me all of the overall steps, but we'll see that here. You go from a 2-carbon molecule back to a 3, to a 4, to a 5. Okay, so it goes through a whole series of steps. Throughout this process of the citric acid cycle, as the acetyl-CoA is converted, you're going to get 2 carbon dioxides. Notice here, you get 2 NADHs at this one part, and then you get an ATP made. We're going to get 1 FADH2 made, and another NADH over here. Okay? This has to go twice. So, from the acetyl-CoA part down, that occurs 2 times. That's where you're getting the 6 NADHs, the 2 ATPs, and the 2 FADs. You get 4 carbon dioxide through the citric acid cycle, and you're going to get 2, because you have 2 pyruvates. You only had 1 up here. You're going to get 2 carbon dioxides. So this is where all the carbon dioxide is being produced. All right. Citric acid cycle has 8 steps. Each has a specific enzyme. The acetyl group from acetyl-CoA is going to join the cycle, so it combines with a 4-carbon compound called oxaloacetate. It's going to form citrate. And then there are 7 more steps that are going to break the citrate back down into the oxaloacetate, which essentially makes it a cycle. And as that cycle occurs, you're going to get the nicotinamide, the NADH, and the FADH2 produced, which will then go to the electron transport chain. Okay, so here's the specifics of the citric acid cycle. So again, this has to occur twice. You don't need to tell me all these steps. I'm just showing you. So acetyl-CoA, you have a 2-carbon molecule. It's going to get added to oxaloacetate, which you see there's a 4-carbon molecule. We get a 6-carbon molecule called citrate. Okay. So between citrate, you're making water, you're using water to convert it to isocitrate. So acetyl-CoA, oxaloacetate, citrate. Okay. Now, during this process, isocitrate is going to be reduced. It's oxidized. The NADH is reduced. You get 1 carbon dioxide released. Okay, so now you've gone from a 6-carbon to a 5-carbon, which is something called alpha ketoglutarate. Okay, same thing. That's going to have another redox reaction. You're going to lose another carbon dioxide. When you do that, you're going to make another NADH. You go from a 5-carbon to a 4-carbon compound called succinyl-CoA. You don't need to know that specifically. Then succinyl-CoA, we're going to actually here you're going to use GTP to help make the ATP. That's going to convert the succinyl-CoA to succinate. Okay, succinate gets converted to something else. As the succinate is converted to fumarate, we're going to make the FADH2. Whoops. And fumarate is going to be converted to malurate, which is going to make another NADH, and then it goes back to the oxaloacetate. Okay. So, again, glycolysis and the citric acid cycle are only going to produce 4 ATP molecules overall through substrate level phosphorylation. Most of the energy that we're going to get is from the NADH and the FADH2, because they're going to carry these electrons down the electron transport chain through oxidative phosphorylation. Whoops, before we go there. So, again, just to make sure you... So, again, pyruvate oxidation, you have 2 pyruvates. At the end of that, you get 2 carbon dioxides, 2 acetyl-CoA, 2 NADH. The acetyl-CoA goes into the citric acid cycle. There's a series of 8 steps. At the end of those 8 steps, you are left with 4 carbon dioxide, 4 NADH pluses, 6, 2 ATP, and 2 FADHs, because remember we had 2 acetyl-CoAs. So whatever you see on this diagram, you have to multiply it by 2. That's what happens by the end of the citric acid cycle. Alright, so the electron transport chain, or ETC, is in the inner membrane of the mitochondria. So in prokaryotic or bacteria cells, this actually just occurs in their regular plasma membrane. Alright, so most of the ETC, or the electron transport chain components, are just a series of proteins that have electron carriers, and we just call them 1, 2, 3, and 4. Most of these proteins are called cytochromes, and they are going to have heme groups. So heme is the same protein in our hemoglobin, so it can carry metal ions, things like that, so that's going to function as the electron carrier. All right. The electron carriers are going to alternate between being reduced and oxidized, because they're constantly going to be giving up electrons and gaining electrons. And every time the electrons get transferred, there's going to be a drop in free energy. So you know, like if you start the slinky at the stop, I don't even know if you guys know what a slinky is. It's this big old spring-looking thing that's supposed to walk downstairs. And back in the 70s, it had a cute little song, Slinky, Slinky, what a wonderful toy. But anyway, so if you have that, as it goes down the stairs, or as a ball bounces down the stairs, you're losing a little bit of energy each time, because eventually you're going to get to the end. So you're going to lose some free energy every time you transfer an electron. By breaking that free energy drop into small, like stepwise reactions, we can release the energy again in manageable amounts, not release it all at once, where you have all this hydrogen ions or hydrogen gas that's then going to be exploding or explosive when we have the heat energy added to it. All right. So in order to reduce the oxygen, we're going to make water. So you get the CO2 is made in the citric acid cycle. You get a little bit of water made. Most of the water that is formed is going to be formed here in the electron transport chain. ATP isn't produced directly by the electron transport chain. It's made by ATP synthase and part of this chemiosmosis. The ATP synthase essentially is like a little proton pump. Think of it as like when you go through, like at a football stadium or through the metro, and you have that little bar thing that you have to push that rotates around. As it rotates and lets a person through, it's pumping a hydrogen through, which then provides the energy to add a phosphate to the ATP. But you'll see that here in a second. All right, so oxidative phosphorylation, this is where we're going to get the majority of the ATP. All right, again, you do not need to know all of these, okay? And most of these complexes are just numbered in the order in which this occurs. That's at 1 to 2 to 3 to 4. All right, so NADH is the least electronegative because it only has one H, so it has 2 electrons. FADH2 is a little bit more electronegative. All right, so you can see here that as we transfer these electrons from the NADH or the FADH2 are going to go through a huge series of carriers. So in protein complex 1, it's FMN, and then it's an iron sulfide. The iron sulfide is going to transfer it to, it can either transfer it directly to this intermediate Q, or you could have the FES take the electrons from FADH2, and then it's going to go through another cytochrome B, another iron complex, cytochrome C1, cytochrome C, cytochrome A. So you don't need to know all those. Just know that electrons are transferred from molecule to molecule to molecule from either NADH or FADH2 through these complexes. And at the end, you have these 2 electrons that are left. Okay, so kind of think of it as maybe if you had a whole plate of cookies and I said, here, take one and pass it down. By the time the plate of cookies got to the end of the row, there wouldn't be as many cookies there, okay? Because each person would take a cookie. All right. So the last carrier, cytochrome A3, is going to pass the electrons to oxygen. Oxygen is pretty electronegative. Now again, oxygen is usually as an O2 molecule. It wants to be there. That's why it has 1/2. Okay, it's going to take 2 hydrogen ions and 1 oxygen to make a water. So this has to happen twice. Right? So the energy that we get released by the electron transfer is going to pump hydrogen ions into the inner membrane space. So every time we pump a hydrogen, because remember we talked about active transport, we're creating a hydrogen gradient across that membrane space, but it's also a chemical electrical gradient because you have all the pluses on one side and you have all the hydrogens on that side. So all the hydrogens are ions. So you have the chemical concentration of the hydrogen, plus you have the electrical charge. So as that hydrogen is going to diffuse across its gradient back into the mitochondria, that's going to help basically cause the rotation of the ATPase synthase enzyme. Okay. Hydrogen can only go into the inner membrane through protein complexes called ATP synthase. So the ATP synthase uses the exergonic flow of the hydrogen to drive the phosphorylation of the ATP. And since the hydrogen ions are essentially diffusing across a membrane from an area of high to low, we're calling this chemoosmosis or chemiosmosis because they're in solution, but the energy from that hydrogen ion gradient is what's driving cellular work. So it's called chemiosmosis. All right. So that energy stored in the hydrogen or proton gradient across the membrane is going to couple with the redox reaction. So basically it's going to drive the redox reactions of the electron transport chain so that we can make ATP synthesis. So we call this a proton motive force, meaning the protons are the motivating or the moving force that's going to give us the capacity to do the work. So if you want to watch the animations of the rotating ATP synthase and the Bioflux animations, and then watch the electron transport chain. And it is really kind of cool. They've actually looked at it, and it really does look almost like a little carousel. So every time a proton goes through there, it does one turn of the ATP synthase molecule. And as it does that, it's going to eventually transfer the electrons to ATP. All right, so it looks like this. It looks like this little carousel pump type thing here. And again, you do not need to know all these steps. I just want you to know the general process. All right, so we're looking at the inner membrane space. You have the mitochondrial matrix, which is basically like the cytoplasm of the mitochondria, okay? So we have these proteins, these integral type proteins. Okay, so you have all these hydrogen ions in the inner membrane space. You have all these little rotors, okay, that have little holes that specifically allow for a hydrogen ion to attach to them. All right, so as a hydrogen ion wants to diffuse, it's going to diffuse through this little purple channel here in the stator, okay? Every time it diffuses, it's going to attach in here to the rotor. Well, when it does that, it changes the shape so that causes the rotor to spin one time. And every time a hydrogen ion makes one complete turn before it leaves the rotor, it'll leave through the secondary channel. Spinning of the rotor is going to cause an internal rod that spins as well. It's going to extend like a little stalk, and when it does that, it has basically some catalytic sites that act like enzymes, and eventually what that will do is it will kind of like cranking, I'm trying to think of the word I'm looking for, but you basically go through a little crank, and at the end, ATP pops out. All right, so the only way I can kind of think of it is you have a carousel. Every time a hydrogen ion goes through there, you do one rotation, like you're cranking an old gear or something, and it causes something to spin, and eventually you get to the end and the ATP comes out. So you're basically forcing the ADP and the phosphate to come together, and you get the ATP. I'm thinking of, in my head, I'm visualizing like when you put, if you ever go to like Hershey or Disney or any of those places where you put in the quarters and the penny, okay, kind of think of it like this. As you put the quarters and the penny in, every time you put the money in, then you start to do the little crank, okay, and it puts the penny down there. And every time you do the crank, it's cranking it closer and closer and kind of squeezing that penny out. And then at the end, the penny pops out with the shape of whatever you picked. Hershey chocolate bar, Reese's cup, Shamu, whatever you do at SeaWorld. So that's how I'm visualizing it in my head, okay? So this is kind of how it works. Make sure you watch those animations. All right, this is just showing you the computer model with the actual ribbons of the 4 pieces of the ATP synthase. So you have a whole bunch of different polypeptides that are going to work in this. We don't know how all of it works, but we do know that some of it rotates. All right, so oxidative phosphorylation is where we're going to get the majority of the ATP. All right, again, I don't want you to know all those 50 million steps and all those different carriers. I just want you to know, basically, the electron transport chain is going to transport the electrons using NADH and FADH2 through a series of these protein carriers. And notice every time something gets reduced, a hydrogen gets shot across one of these protein carriers, so, except number 2. Number 2 it doesn't. So every time you're getting an electron and you're getting the hydrogen ion, we shoot the hydrogen ion across into the intermembrane space so that we have this huge concentration of hydrogen ions. Eventually, the hydrogen ions are going to migrate down here to the little ATP synthase molecule. Okay, now you have these 2 electrons at the end that are just floating around. You don't want them shorting anything out. So they, along with 2 hydrogens that stay behind, are going to join up and make water. Okay, and then all those hydrogens essentially are going to start to go down and cause this little ATP synthase through chemiosmosis to rotate. And every time a hydrogen ion comes in, it causes one little rotation of the, think of it when you're putting a quarter in and you're trying to get a gumball or something at the store, okay? You have to turn it several times before you get something out, okay? So that hydrogen, once it gets in the seat, or if you think of getting on a Ferris wheel, when you want to get off the Ferris wheel, he's got to go all the way around and stop at each seat, right? So you go all the way around once, and then he starts letting people off. That's how these hydrogens get in through here. Once that hydrogen comes all the way through the bottom, it's going to power that hydrogen ion, the electrical charge, basically, that power is what's going to add that phosphate back to the ADP and essentially recharge it to become ATP. All right. So here's how we're going to account for everything that happens in cell respiration. Most of our energy is going to flow this way. Glucose to NADH, to the electron transport chain, to the proton motive force of chemiosmosis to ATP. What did we say? You have 6. About 34% of the energy in the glucose molecules transferred to ATP. So we make about 32 ATP here. Remaining energy from glucose is lost as heat. Okay. I can't remember the exact numbers. I want to say 6 times 3 is 18. You end up making, it's like 2 or 3 for each NADH and 2, I think, for the each FADH2. You don't need to know that. But in theory, at the end of chemiosmosis and the electron transport chain, that's an additional 32 ATP from the 2 that we made in glycolysis and what we made in the citric acid cycle. Okay. So again, the exact number of ATP molecules that we make is not really known. I've seen it again anywhere in a book that it'll say the overall average is anywhere from 30 to 38. Phosphorylation isn't directly coupled to the redox reaction, so it's not a one-for-one transfer. ATP yields are going to vary depending on whether the electrons were carried by the NAD plus or the FAD. And then again, that proton motive force that generates the electron transport chain is also used for other kinds of work. So it also depends on oxygen concentration, if the hydrogen ions are there, pH, temperature, all that type of stuff. So make sure you watch the animations that talk about the factors that affect ATP yield and ATP yield from cell respiration. All right. For our purposes, though, we're going to say that you get 2 ATP overall from glycolysis. Okay. We get 2 ATP from the citric acid cycle. Now your book here has it low. It says 26 to 28. Most books that I have seen in older books will say 30 to 32. So that when you add those all up together, your book is saying 30 to 32 ATP. Again, I've seen it. Typically, books will tell you the magic number is 36. So just realize if you're looking at something else, or if you happen to see a test question that I pulled from a different test bank and it says 36, just realize there's a range, okay? So again, we get 2 ATP at the end of glycolysis. We had to add 2, but we made 4, so we're up 2. Your NADHs are either going to shuttle it then to 2 NADHs or 2 FADHs with the pyruvates. Okay. At the end of the citric acid cycle, we now have had 6 NADHs plus 2 FADH2s. We have 2 more ATP. And then all those, the 2 NADAs or the 2 FADHs plus the 2 NADs from the pyruvate and then the 6 NAD FAD here are going to go into phosphorylation. And again, you're going to get about 26 or 28 ATP depending on what it says there. Hold on, I'm going to... pause this for a second while I try to find a different picture for you. So hold on just a second. All right, hopefully you can see this. For each NADH, you get 2 or 3 depending again on other factors. FADH2, you get 1 or 2 ATP. So overall, this is kind of what I want you to be able to tell me here. Stage 1, again, is glycolysis. So look here, we put 2 ATP in, we got 4 ATP out, okay? So you're only got 2 overall. We get 2 NADHs, we get 2 3-carbon pyruvates. Remember, this is going to go through the pyruvate conversion to acetyl-CoA, okay? When that occurs, you get 2 carbon dioxide. It goes into the Krebs or the citric acid cycle. At the end of the citric acid cycle, which is stage 2, we've had an additional 4 carbon dioxide, 2 ATP. You had 2 NADHs here, 8 NADHs here, 2 FADHs here. So now this will say 6. Some books will say 6 here. So this then will tell you, then through oxidative phosphorylation, you could get up to 34. So you can see the changes and the variety in books. Typically the magic number that I use, just because that's how I've known it for the last 25 years, has been 34 to 36 ATP. Okay, so just wanted you to be aware of that in case we saw that. All right, so just real quick again. Oh, this is going into fermentation, so that's something else. I want to, go back. I want to end there so that we can stop the show so it's not that long, and I'll finish up this chapter with fermentation here